Hello guys, my name is Alfredo Garcia and on behalf of Grand Canyon University, I'm going to be presenting glycolysis, gluconeogenesis, and the role of the enzyme aldolase. So, glycolysis, simply put, is the breaking down of glucose inside the cells, and glucose, of course, is one of the most important sugars that we rely on for energy. So this is the process which allows cells to extract or produce energy in the form of ATP, or in order to create ATP. It's an anaerobic process, which means that it doesn't require oxygen. It takes place in the cytoplasm of cells, and it requires a kickstart in energy. So cells have to rely on a little bit of input of energy in the form of ATP before they can get this process kickstarted. So this is what the beginning of glycolysis looks like. So it's somewhat complicated. There's a lot of steps, but all that these steps really are intermediate. So you're breaking down glucose, and you get all these intermediates and all these enzymes which function to make these intermediates. So I'm kind of gonna go over them as best as I can. So the very first step, if you guys look here, um, hexokinase catalyzes the reaction between glucose and ATP, phosphorylating the molecule. And phosphorylating just means adding a phosphate group or a phosphorus. So from that, instead of having glucose, you end up with glucose 6-phosphate. And after that, the glucose molecule can't leave the cell. So once glucose is phosphorylated, it's stuck inside the cell because it can't leave through the membrane channels that it can come and go through. So from there, after glucose is phosphorylated, you get an isomerase, so called phosphoglucose isomerase, sorry. And what an isomerase is just an enzyme which creates a different isomer of the molecule. So an isomer is just where you have the same chemical formula, but the atoms are kind of mixed around, so they're in a different structure. So this enzyme converts glucose 6-phosphate into the isomer fructose 6-phosphate, which you guys can see here. So in step 3, the enzyme phosphofructokinase catalyzes the phosphorylation of fructose 6-phosphate. So the isomer now is phosphorylated again. So you end up with 2 phosphate atoms in the molecule. So once again, ATP is used as the phosphorus source, and you get fructose 1,6 bi or diphosphate. And this addition of phosphorus makes glucose unstable. So the more phosphoruses that you add, the more unstable glucose gets, which is a good thing because that means that it will be easier to break apart. So step 4, aldolase, which is the important enzyme that we're going to be talking about here, cleaves fructose 1,6 bi or diphosphate into the 2 3 carbon isomers, dihydroxyacetone phosphate and um, glyceraldehyde 3-phosphate. So these are 2 isomers, and we want to end up with 2 of the same molecule. So the last step is that a different isomerase transforms dihydroxyacetone phosphate into the isomer glyceraldehyde 3-phosphate, producing 2 glyceraldehyde 3-phosphate molecules overall. So now we've sort of broken down glucose, and we have 2 of these glyceraldehyde 3-phosphate molecules. So in step 6, the sugars produced in steps 4 and 5 are oxidized, and the electrons are picked up by NAD+. So the sugar is then phosphorylated again, producing 1,3-biphosphoglycerate. So for step 7, phosphoglycerate kinase allows 1,3-biphosphoglycerate to donate a phosphate group to ADP, creating 3-phosphoglycerate. And in step 8, 3-phosphoglycerate is converted into a different isomer, 2-phosphoglycerate by a different isomerase known as mutase. So then the last two steps, step 9, the enzyme enolase allows for the dehydration reaction of the 2-phosphoglycerate, which converts it into phosphoenolpyruvate. And this molecule has a higher energy phosphate bond. So step 8, the enzyme pyruvate kinase allows for substrate level phosphorylation of ADP, again producing the molecule pyruvate. So this last step is one of the most important steps in glycolysis, because when you get this substrate level phosphorylation, what that is that you're getting a molecule of ADP, so adenine diphosphate, and you're removing a phosphorus or phosphate group from one of these molecules and adding it back to ATP, which can then be used again as a form of energy by the cell. So gluconeogenesis is a different process. It's very similar to glycolysis, but somewhat in the reverse. So it produces sugars, glucose, and it's important for cells in the brain, testes, blood, and kidney. So this is a process which occurs in the liver and kidney, and it involves 3 different enzymes. So we said it's kind of the reverse of glycolysis, but there's 3 subtle differences. There's 3 steps that are going to use 3 different enzymes, and those 3 enzymes which are going to be replaced are hexokinase, phosphofructokinase, and pyruvate kinase. So these are replaced by pyruvate carboxylase, PEP carboxykinase, and malate dehydrogenase. So if you guys want to look at it here, this is a useful diagram, but it doesn't really help us make sense of it. So as a standalone, it's good, but it's a lot better if you can compare the 2 processes together. So if you look here, you'll see that for the most part, the 2 processes are very similar. You're going to see that a lot of the steps are just in reverse, and you're going to be using the same enzyme. So for example, with glycolysis, you start with glucose and you end up with 2 pyruvates. With gluconeogenesis, you start with 2 pyruvates and you end up with glucose. And you see a lot of the same enzymes. So for example, here, step 2, phosphohexose isomerase. Here on the other side with gluconeogenesis, phosphohexose isomerase. Here, aldolase, which we said is the important enzyme we're going to be talking about. Triose phosphate isomerase, so a different isomerase that's the same for both of them, but then there are differences. So for example, here, step 3, phosphofructokinase 1 for glycolysis, and fructose 1,6 biphosphatase for gluconeogenesis. So let's talk about the enzyme aldolase. So aldolase is one of the many important enzymes involved in catalyzing the reactions that occur in converting glucose into pyruvate and pyruvate into glucose. So if we go back and look at this, and like I mentioned, you're going to see aldolase in both reactions. So aldolase allows for both of these reactions to happen. So there's some differences, like we said. So aldolase, fructose biphosphate A. Sorry, disregard that. So aldolase functions via lysine and tyrosine residues, and where tyrosine acts as a hydrogen acceptor and lysine stabilizes the intermediate via covalent bonds. So why is this enzyme important? So it's important physiologically as it catalyzes 2 of the most important reactions in the human body. So these 2 reactions are some of the most important reactions that happen inside of our cells. So glycolysis allows us to break down the simple sugar glucose and derive energy from it. So it's not the only means that we have of deriving energy, but it is a very important part of that process. With gluconeogenesis, producing glucose is important because a lot of the cells inside of our body have to rely on glucose. So they can't rely on other things for energy. They have to break down glucose. So in instances where glucose might not be available, these cells need to have a means of making this glucose from other reactions and other molecules. So deficiency of this enzyme causes hemolytic anemia due to the difficulty in producing glucose. And this is an autosomal recessive disorder. So we somewhat talked about this briefly a little while ago, but glucose is very important for blood cells. So blood cells are some of these cells which rely mainly or only on glucose as a form of energy. So if you have a deficiency of aldolase, then you can't really go through the process of gluconeogenesis, or this process is going to be very, very hindered. And this, of course, is going to cause blood cells to die off, right? So this dying off of blood cells is what causes this hemolytic anemia. So hemolytic anemia is where you have less blood cells than what is optimal. So overexpression of this enzyme is possibly associated with tumor progression, and this is based on correlation between colon cancer progression and upregulation of the gene. So this is something interesting that we can talk about, but there isn't a direct correlation between these two things. So we don't have any current research that says that there is a very specific cause or connection between overexpression of aldolase and tumor progression, but there is a correlation there. So we don't know if the two are connected, but it has been noted that when there is a large amount of tumor progression, then usually aldolase has been overexpressed, or there's an overexpression of aldolase. And glycolysis is possibly associated with cancer progression. So this is something else that has a lot of evidence pointing in that direction. So there are a lot of research papers and research that points at the possibility that this process of glycolysis is associated with cancer progression. And there's not really a simple cause for this, or very very a well-known mechanism or path that leads to this, but it would make sense, seeing as cancer is uncontrolled cell division, and when cells are growing they require energy. So if you need energy, you need to be creating a lot of glucose. So it definitely makes sense that there would be some connection between glycolysis, breaking down sugar for energy, and your cells dividing uncontrollably.